Today we're going to talk about protein synthesis. What is protein synthesis? Well, if we look at these two words, it's the developing and building of proteins from smaller structures to a larger, more complex structure. But how is this done? Well, let's first take a big look at the cell. If we look closely at this cell right here, we will see that it has many different parts. But the main part we want to look at is right there in the nucleus of the cell. In this nucleus, we will find that we have something called DNA. Right there in the nucleus. Now, DNA is known as deoxyribonucleic acid. It is one of the two different types of nucleic acids that we tend to see inside the cell. It is made in a unique structure known as a double helixed structure. It looks kind of like a twisted ladder. Now, what is it composed of? Well, it's going to have a phosphate group on the outside. These are making the actual outside portions of the ladder. Then it has deoxy or deoxys that is known as the D portion in this picture, and these are what attach to the phosphate groups there. Now, what attaches to the deoxys is what we call nucleotides. Now, there are four different basic types of nucleotides: adenine, guanine, thymine, and cytosine. These are the four different types of nucleotides. These nucleotides act as parts of a code, the same way the letters of the alphabet, when placed in a specific order, create words. When these guys are placed in specific orders, they create brand new bits and pieces of information. Now, only certain nucleotides will match with other nucleotides, which is very important in the next stage of what we're going to talk about. What this means is that all nucleotides are matched only according to one other certain partner. Specifically, adenine will only match with thymine, and vice versa, thymine will only match with adenine. Guanine is only going to match with cytosine, and at the same time, cytosine only with guanine. The order of the nucleotides determines the message that the DNA is trying to send. Now, this is known as the genetic code. The different patterns that this code makes determines hair color, eyes, shape of your body, size, weight, all the things that make you what you actually are. It is this code also which holds the blueprints for building proteins. And proteins, if you remember, are the building blocks to all parts of the cell. Now, there is still one problem. Where is the DNA located? Well, it's found in the nucleus of the cell, which is in the center of the cell. Where are the proteins built by the cell? Well, they're built in ribosomes, which are found on the outside of the nucleus. How then does the information get from the nucleus of the cell to the ribosomes? Well, the answer is in the process of transcription and translation. RNA is the other nucleic acid that we deal with. RNA is known as ribonucleic acid, and it's slightly different from DNA. It has almost the same code as DNA, aside from one individual nucleotide. In place of thymine, it actually has something known as uracil. Now, transcription. The word transcription actually means to convert one type of writing into another, or one form of the same language into another form of that same language. For instance, just like a secretary might take notes using a tape recorder and then turns around and transcribes them into written text. That is a form of transcription. The DNA does something similar to that. This involves the transfer of information from DNA's base sequence into a complementary RNA base sequence. Specifically, type of RNA that we're going to talk about here is known as mRNA. The M stands for messenger, and that's because it's actually going to deliver a message. Now, translation, on the other hand, is literally dealing with language. Translation is taking words in one language and restating them in another language entirely. For instance, like someone translating from English to Spanish. Now, what does this mean? This means that the language of the nucleic acid base pairs is translated into the language of proteins. Now let's look at this diagram right here. What I want to do is take us through the very different stages of transcription all the way to the point where we are actually performing translation. Well, the first stage that I would like to look at is right here. This is known as transcription. Here, if you look over here, mRNA is doing what we call specifying one polypeptide and is made on a DNA template. Now, let's put that into layman terms. What's happening here is this RNA, that's this blue strand right here, is matching up with an unzipped DNA. Now remember, we said it's a double-stranded helix, okay, or a twisted ladder shape that we have with this DNA. Well, what happens right prior to transcription is the DNA will actually have its bonds between the nucleotides broken only for a temporary time. And what happens is it literally unzips like a zipper. And 1/2 of that strand is exposed to the RNA. And here we actually see the code, AATCGTTCGCTA. That's a big long code right there, and these codes can go a lot longer if you can even imagine this drawing trickling off way off into the distance. But what happens is this RNA is going to begin to build and match up according to the specific code that the DNA is already revealing. Now, remember I told you that RNA doesn't have thymine. And thymine is the one part of the DNA that or the one nucleotide of the DNA that matches up with adenine. But what RNA does have is uracil. So if you look right here, you're going to see that everywhere that we would normally have a thymine matching up with the adenine, it's a uracil instead. And that RNA will match up all the way down that split area of the DNA code until it is completely made a brand new RNA, or specifically in this case, mRNA molecule. Now, the next stage is that RNA molecule will actually leave the DNA, or it will leave the nucleus of the cell. mRNA leaves the nucleus and attaches to a ribosome, and translation now begins. That whole process that we just explained was transcription. Now we're going to attach to the ribosome. This is stage 3. Stage 3 is where once this ribosome latches on to this RNA, it's going to begin to siphon it through, almost kind of like a typewriter, pushing things through, or as it moves down, it actually starts to match up the code. But what is it matching up with? Well, we have something called tRNA. tRNA recognizes a complementary mRNA codon, calling for its amino acid by building via its anticodon to the codon. Okay, anticodon to the codon. That's all confusing language. Make it simple. This right here, this pink structure, is known as an anticodon. This structure right here on the mRNA is known as a codon. Now, if you think about it, anti means against or opposite of. Well, we know that certain pairs only match together. They're the opposite of the other pairs. In this particular case, these anticodons are going to have 3 nucleotides attached to them. Still, uracil is going to replace the thymine. Okay, so the uracil, it will match up every time you see an adenine in this mRNA. You'll see a uracil on the anticodon of the tRNA. Adenine will always match up with an opposing uracil on the mRNA. Now let's look at this little structure right here that's attached to the top of the anticodon. That is very important for us to recognize as an amino acid. That amino acid is the beginning or the basic component that is going to make up what this whole chain of amino acids is known as a protein. Now, we call this a polypeptide chain. Why is it called a polypeptide chain? Simply, these amino acids are attached to each other by a peptide bond. So what's happening here is this larger protein, known as a ribosome, is providing an attachment point or place where this particular amino acid can react with these other amino acids. But only the specific amino acid attached to the correctly coded anticodon is going to be used in the correct sequence or the correct order as controlled by the ribosome, which was ultimately dictated by the mRNA that got its information from the DNA of the cell. So what happens here is this anticodon is attached to the ribosome. The ribosome then allows it to latch onto these other nucleotides found on the mRNA according to the correct code. Then when this happens, just like right here, that amino acid that is attached to that anticodon is then going to form a polypeptide or a peptide bond with the adjacent or the amino acid right next to it. And then the anticodon is going to release that amino acid, and it is actually going to be released by the ribosome to go back out and attach to another amino acid of the same type. So as ribosomes move along the mRNA, a new amino acid is added to the growing protein chain. What that means is then the released tRNA re-enters the cytoplasmic pool, and it is getting ready to be recharged with a brand new amino acid. Now, this is how this structure is moving. It's moving gradually in this direction. So as it moves on, new proteins are built, or a protein is continuously being built until the mRNA is completely pushed through the ribosome. Once that protein is built, it is then going to be transported away from the ribosome through the endoplasmic reticulum to the Golgi complex, or also known as the Golgi apparatus, where then it will be put to work or outfitted with a